Alright, so we went through some parts of Chapter 4 in class, and I just wanted to provide you with another run-through of the material to help you as you try to study Chapter 4, which is a tour of the cell. So we began by talking about the tools that biologists use to study cells, and the most important tool that we talk about in class is the microscope. So you guys had the opportunity to use microscopes in class, and in fact you would have used light microscopes, which allow you to have visible light passing through a specimen and through lenses to make a bigger picture of what you're looking at. The important thing I need you guys to know about light microscopy is that light microscopy is pretty good up to a limit. And that limit is about 0.2 microns, which is the point in time when you can no longer resolve structures or samples. You can't distinguish them from each other. That is 0.2 microns to be up here. And the problem becomes if structures or cells are smaller than that, you can no longer observe them. And so what you would use in that situation would be an electron microscope. So the big difference between an electron microscope and a light microscope, other than the fact that you can observe a much smaller thing with an electron microscope, is that an electron microscope uses electrons as opposed to visible light to observe, to make visualizations of samples. And so you are using sort of a different functionality to see things that are even smaller, and you can notice on this graph you can get down to protein visualization with the right type of electron microscopy. This slide just shows you a bunch of different samples of microscopy. Light microscopy up here on top. This should look like the types of things that you saw in class. Fluorescent microscopy we touched on briefly. That just allows you to visualize specific structures within a cell. So it's important on fluorescent microscopy to note that the green is one protein, the red is another protein, and the blue is a third. In this case, not a protein, it's DNA, but it lets you identify specific things inside of the cell. Finally, you can see in electron microscopy we get much better resolution at a much smaller size. And we have two kinds, scanning electron microscopy, which looks like a 3D image, and transmission electron microscopy, which is going to go through a sample and is going to let you see the interior of that sample. So when we think about cells, we want to think about the big things that make them all identical, what makes them all similar. So all cells have a plasma membrane, all cells have a cytosol, all cells carry genes, they have chromosomes or DNA, and all cells have ribosomes, they can make proteins. And we're going to think about this in terms of what makes the basic features of the cell become more complicated to get to what we call a eukaryotic cell that has internal membranes and compartments. So this is your exemplar of a prokaryotic cell, and you'll notice that a prokaryotic cell has no nucleus. It just has its bacterial chromosome free-floating in the cytosol, and we call the region where it floats the nucleoid because that just identifies where we're seeing the DNA. Some other structures on the prokaryote that you should be familiar with, the ribosomes, which make protein, the plasma membrane, which surrounds the entire cell and keeps it bound. We have a cell wall, which is a structural protective feature of the prokaryotic cell, the capsule, which is a polysaccharide coating on the bacterial cell that helps it stick, avoid the immune system, things like that. Flagella help with movement, and fimbriae or pili help with attachment to things inside of the environment that the cell lives in. So you can see this is a very simple cell. It doesn't have a whole lot of things going on with it. And we can compare that to a eukaryotic cell. And so if you look at the difference here, right, very few things. It has just those four things we mentioned all cells have to have. And then when we get to a eukaryotic cell, we start seeing all kinds of stuff. We have big nucleus that contains the DNA. We have all sorts of endomembrane system, and we'll talk about what that does. We have organelles that make energy, organelles that cause the cell cytoskeleton to be supported. There's a lot of things going on, a lot more compartments. By adding more compartments to the eukaryotic cell, you can actually increase the complexity of the cell and make it a much more complicated processes happening in the cell. Much more things can go on in this cell. So at the outside of both the prokaryotic cell and the eukaryotic cell, we have the plasma membrane. And so the next chapter in our book will go into much more depth about the plasma membrane, but for what you need to know now, the plasma membrane is primarily composed of phospholipids. You see these phospholipids here. They have a hydrophobic tail that faces in, 2 hydrophobic tails and a bilayer, and a hydrophilic head that faces out. So the exterior of the cell is aqueous. The interior of the cell is aqueous, so the hydrophilic heads face those aqueous portions, and then you have a layer of hydrophobic that prevents anything from crossing through this membrane, with the exception of proteins that can go through this region and make channels for items to go in and out. And again, we will cover this more in the next chapter, but for what you need to know now is we surround the entire cell with a semipermeable membrane, and that membrane can let certain things in and keep certain things out. And that's really important for the cell, that it's able to define its interior and control what goes in and out of that cell. And speaking of things going in and out of the cell, the surface area of the cell membrane, of the plasma membrane, really impacts how many things can get in and out of the cell, and specifically think about how much energy or nutrition can get into the cell. So if we have a small cell like a prokaryote, this cube with sides of length 1 will have a surface area of 6, so that's the height times the width of all the boxes times the number of sides. It's going to have a volume of 1, so its surface volume ratio is going to be 6. Now if we make this cell, this cube, bigger, right, we have its sides of 5 now, the surface area increases, but so does the volume. And so if you think about this bigger cube, it has a surface volume ratio of 1.2. And if you think about this cell needing to get sugars, for example, into the cell, it doesn't have nearly as much plasma membrane to allow transport in and out relative to this much smaller cell. And so what you would need to do when you increase in surface size is you have to add more membrane structure to increase the surface area of the cell, and this would be the distinction between a prokaryote, which is limited by the fact that it has no interior membranes, as you approach a eukaryote that can become larger because of its specialization, and eventually to a multicellular organism like us that takes advantage of the fact that lots of cells means lots of surface area relative to the volume in each individual cell. So again, here's our classic eukaryotic cell. This happens to be an animal cell. And this would be another eukaryotic cell. This would be a plant cell. And so some of the features that you should notice that are different between the two is that the plant cell in particular has a cell wall, which the eukaryotic animal cell does not. And the plant cell has chloroplasts, which would appear to be green in light microscopy. And the final thing that your textbook is going to tell you is unique to plant cells, and this particular structure is unique, the central vacuole, which is a big storage structure. But I don't want you to think that animal cells can't have vacuoles, because they can. They're just not going to have this large central vacuole that takes up a large portion of space inside of the cell. All right, so starting in on a tour of the particular organelles, let's begin at the center of the cell, the nucleus of the cell, where the genetic instructions are housed, and then those genetic instructions are carried out by the nucleus. So in a eukaryotic cell, all of our DNA is going to be put into the nucleus. And the purpose of putting the DNA in the nucleus is it provides it some localization so that the cell knows where it is, and also some protection, because that DNA, which contains all of the instructions for how the cell should function, is pretty important material for the cell. So you'll notice that it goes inside of a double membraned sac. So the nucleus has an inner membrane and an outer membrane. And importantly, this membrane does have pores that go through it. And the reason you're going to need pores to go through the nucleus is that you're going to want to allow certain proteins in so that they can tell the DNA to be copied and things like that. And you need to allow mRNA to come out so that mRNA can be utilized to make proteins. As well as letting mRNA out, you're going to have to also let ribosomes out of the nucleus, because ribosomes are formed here in the nucleolus region of the nucleus. So the nucleus contains all of the DNA, which is packed tightly in structures called chromatin. That chromatin kind of fills the entire sac of membrane, and as it gets copied, portions of mRNA that are copied from it are able to exit the cell and do other things inside of the cell. So if we zoom in now on a ribosome, as we said, was made in the nucleolus. A ribosome is actually composed of RNA and protein, so that's why it's made in the nucleus. And it's composed of two pieces, a large subunit and a small subunit. We will cover with more specificity what exactly the ribosome does in a future chapter. But for now, just know that the ribosome is able to attach to a piece of mRNA, so nucleic acid, and turn it into a protein by reading along that mRNA and putting the appropriate amino acids in the appropriate order to make a protein for the cell. So ribosomes are critical for both prokaryotic and eukaryotic cells, because if you can't make proteins, the cell can do nothing important. So speaking of proteins, we will then talk about the endomembrane system. So the endomembrane system is what allows proteins to be created, trafficked throughout the cell, processed throughout the cell, and ultimately put in their final locations. So we will actually start, our definition of the endomembrane system at the nuclear envelope. And we can think of this going in order from the nuclear envelope to the endoplasmic reticulum, to the Golgi apparatus, and then from the Golgi apparatus you basically have three places you can send a protein. You can send it to a lysosome to be destroyed in case you didn't like what you made. You can send it to a vacuole to be stored for later. And you can send it to a plasma membrane so that you can either release it from the cell or at the very least put that protein into the plasma membrane so that you can have some functionality at the membrane. Between all of these features of the endomembrane system, components are either continuous, so they're connected to each other, or proteins transfer by vesicles, which are small membrane sacs that can move between the structures. So we will start at the endoplasmic reticulum. At the endoplasmic reticulum, you have two types of endoplasmic reticulum, the smooth ER and the rough ER. The major structural difference is that the rough ER is coated with ribosomes. That allows proteins to be made at those ribosomes on the surface of the ER, and the proteins can be either put into the endoplasmic reticulum or put into the membrane of the endoplasmic reticulum. The smooth ER has no ribosomes, so the smooth ER's function is not really to make proteins. It's more relevant for making lipids that the cell will need for later, making those vesicles that the endomembrane system can move throughout the cell. So as a protein goes through the ER, it will be processed, it will be folded, and it will eventually be dumped into a transport vesicle. And the next step after the ER is that transport vesicle will go to the Golgi apparatus. Now you can see the cis space, the receiving side of the Golgi, receives that vesicle. Proteins are then processed through the Golgi in various steps, and they are released from the transface in another vesicle. So you can imagine this as a protein coming in, it gets processed, and then released on the other side. So we go, two steps here. We go from the ER, where the protein is first made. It's folded as it moves through the ER. When it reaches the end of the ER, it will hit the Golgi apparatus, be processed even further, and then released from the transface of that Golgi apparatus. In a second video, I will talk about what the lysosome does, and I want you to move to that next video when you're ready.